Very briefly, I'm going to talk about very briefly today on the bactericidal performance of some new nanostructured composite coatings that we have made in order to destroy Pseudomonas aeruginosa. So this work was done in collaboration with, partly with Dr. Mark Peplow over in the medical microbiology department, and unfortunately he has retired, so we're always looking for new collaborators as we go on through the presentation today. So just to give you some brief overview, we do already know for a fact that titania as a material does possess some photocatalytic activity, and that is pretty much the ability to produce oxide species or reactive hydroxyl species that can be used to destroy a variety of different organic molecules that may be found in bacteria, or just simply to react with organic molecules that are found in a variety of different scenarios. This typically occurs as a result of shining some kind of light or some kind of radiation onto the titania surface in order to promote this type of photocatalysis, or the destruction or the reaction with other materials. Now, it has been found that very, very thin TiO2 films can be used for photodegradation of materials such as E. coli and even Pseudomonas aeruginosa. But the problem is that we will eventually need material layers that can actually withstand, constant stresses due to cleaning or constant wearing as a result of simply the cleaning process, if we were ever to use this type of material, say in a hospital setting or in a food prep setting. So let me just kind of give you a brief overview of the idea here. If you consider steel as a material that is used somewhat ubiquitously within the healthcare and the food prep industries, bacteria tends to grow very nicely on those types of surfaces. Now, if we can possibly make a layer on that surface that can withstand constant cleaning stresses, have really nice bond strengths to the steel, we can polish that coating, we could make it look like steel in terms of its lustrousness and its appearance, then we might actually be able to have a surface that looks like steel that can actually kill bacteria. And that is the objective of what we're trying to do here, but looking at it more from an industrial perspective. But we'll still talk a little bit about some more of the fundamentals though. So we do know that in thermal spraying there is a process known as high velocity oxyfuel spraying, where we can actually deposit nano-based titania powders. So a nano-based titania powders is one in which I take a 50 to 100 nanometer size particle, spray dry agglomerate it to make a micron size particle that I can then put into my torch, which I'll show in a moment, melt it, accelerate those particles, and then make a very thin coating that has a very good bond strength onto the surface to which I'm depositing, and that will have really good wear resistance against a variety of different applications. So Lima and Marple at the National Research Council in Boucherville, Quebec, actually show that this type of nano-based coating made by HVOF has superior wear performance or wear resistance in comparison to non-nanostructured type powder coatings. And some research from Yang has shown that HVOF spray TiO2 coatings can actually photodegrade phenol. Now, the issue that I'm having with HVOF is that it is very expensive when one thinks about HVOF. You need a footprint that is very large to have the kerosene and the oxygen and the torch. It gets very noisy. You're talking a fair amount of money here. So my hypothesis is, why can't we use a cheaper form of thermal spraying known as flame spraying, which is just an oxyacetylene torch. It's kind of like a welding torch, and you only need a small footprint in order to get it done, a small robot, and we can maybe make some of these same nano TiO2 coatings in order to photodegrade this very complex Pseudomonas aeruginosa bacteria. So that is my hypothesis, and that's what I want to talk about today. But before we do that, let's talk about why Pseudomonas aeruginosa. Why did I choose that to study? Well, from my research and from the research of my students, we've found that Pseudomonas aeruginosa is an opportunistic pathogen. It does compromise or causes problem in immunocompromised individuals with AIDS, cystic fibrosis, those living in healthcare facilities and so forth in terms of the aged and so forth. Now, Pseudomonas aeruginosa is definitely more complex than E. coli because it does have a very thick, nice protein-based biofilm that it uses to protect itself. And it binds very, very easily to stainless steel, which again is ubiquitous within the healthcare and the food industries. So here we see the importance of hopefully making a nice coating that when we polish it will look like steel and then can still destroy complex bacteria or maybe even viruses also. So I mentioned some of the points before as to why we would like to work with Pseudomonas aeruginosa and why we would like, what is our motivation. But after mentioning all of these things in terms of the fact that stainless steel is used again very ubiquitously within the industry, we would then like to make a coating that has very high strength to the steel, will have the photocatalytic bactericidal activity that we need in order to kill some of these bacteria and again even viruses, and us also be able to resist constant wearing as a result of the cleaning processes to which these coatings will be exposed. So our objectives in the work and in this presentation today is just to briefly talk about the deposition and the fabrication of a new type of copper nanostructured titania-based composite coating that was deposited by this more inexpensive process of flame spraying, and then to see if we can combine just a small weight percentage of copper into our TiO2, will we be able to improve significantly the destructive effects or the bactericidal effects of this TiO2 coating. So a little bit of a background on the thermal spraying process. So in terms of what we did with regards to the experimental methods, we took our nanostructured TiO2 powders, which is just an agglomeration of nanoparticles, blended it mechanically with some copper powders. The size of the particles were about 18 microns to about 38 microns overall. We only put 5% copper in the mixture in order to increase the bactericidal effects a bit. And then we use this flame spray torch from Medco to melt the powders, accelerate them, and deposit them. And we used 1010 low carbon steel. We could have used stainless steel also. This low carbon steel was just more available for us. Could have used stainless steel if we wanted to. And our coating thickness was only about 300 microns in the end, about 12 mils in the end. We probably would have added another 10 mils to that for the purposes of polishing in order to finish off the coating. But again, this was an experimental work, and we started off with 300 microns just to see what we would be able to do. So in terms of the flame spraying process, what I'd like to show you here is the actual torch mounted upon a robot, and the red line that I'm showing you here is the acetylene line. The green line would be the oxygen line. I promote or I start up a combustion process to make this high temperature flame, temperature on the order of about 2,000, 2,500 degrees C. And if I wish, I may push some air into the flame to cool it down a little bit, or to ensure that I have complete combustion to increase my temperatures a bit more. And I have a line here for the nanostructured powder or the copper titania powder blend that I inserted into the flame. The particles will move at some very high velocity, impact upon my 1010 low carbon steel or stainless steel substrates, and I will be able to make a very thin coating that I can then polish and give it some lustrousness. In terms of the biological tests that we did start off with a couple of small wells. We had some samples of PAC in brain heart infusion 1.5% agar mixtures that we then put on agar plates. And we did use phosphate buffer saline solution in order to prep the solution to have about 10 to the 4 cells per 100 microliters of solution. We did work with the copper titania composite surfaces after we made our coating. We also worked with some stainless steel surfaces as controls and a pure copper surface as control, just for the purposes of comparison. And then, of course, in order to get some photocatalytic activity going from the TiO2, we then shone regular white fluorescent light on the surfaces. Nothing fancy, nothing expensive, just simple type of things that we would expect that someone can do in their setting, and this is just to show some support for potential commercialization of what we're trying to show here.